Good morning, everyone. Today we'll discuss one of the most fascinating molecules in immunology, antibodies, also called immunoglobulins. These are the key players in our humoral immune response. But first, tell me, what comes to your mind when you hear the word antibody?
Sir, antibodies are proteins that fight infections, right?
Absolutely, but there's much more to them. Let's break it down systematically. Antibodies are Y-shaped glycoproteins produced by plasma cells, which are differentiated B cells. Their basic structure consists of 1-2 heavy chains. These are the longer polypeptide chains. 2 light chains, the shorter polypeptide chains. 3 variable regions, the tips of the Y that bind antigens. 4 constant regions, the base of the Y that mediates effector functions.
Sir, why are there variable and constant regions?
Excellent question. The variable region is unique for each antibody, allowing it to bind specifically to an antigen. The constant region determines the antibody's class and function, like whether it activates complement or binds to immune cells. Now let's dive into the different classes of antibodies, also known as isotypes. Each class plays a unique role in the immune response. There are 5 major antibody classes, each with distinct roles. First, we have IgG, the most abundant, making up 80% of serum antibodies. It crosses the placenta and neutralizes toxins. Next is IgM, the first antibody in a primary response, pentameric, and excellent at complement activation. Then there's IgA, found in secretions like tears, saliva, and breast milk. It protects mucosal surfaces. IgE binds to mast cells and is involved in allergies and parasitic infections. Finally, IgD functions as a B-cell receptor.
Why does IgM appear first in infections?
Because it's secreted early by plasma cells during the primary immune response. Later, class switching occurs, leading to IgG production. Because it's secreted early by plasma cells during the primary immune response. Later, class switching occurs, leading to IgG production. Antibodies bind antigens via non-covalent interactions like hydrogen bonds and van der Waals forces. The strength of binding is called affinity. The Fab, or fragment antigen-binding region, recognizes the antigen, while the Fc, or fragment crystallizable region, interacts with immune cells.
How does this help in immunity?
Great point. Antibodies neutralize pathogens, opsonize them for phagocytosis, activate complement, and trigger ADCC, or antibody-dependent cellular cytotoxicity. Great point. Antibodies neutralize pathogens, opsonize them for phagocytosis, activate complement, and trigger ADCC, or antibody-dependent cellular cytotoxicity. Antibodies are crucial in our vaccines, which induce protective antibodies. Autoimmune diseases where autoantibodies attack self-tissues. Diagnostics such as ELISA and monoclonal antibodies in therapy.
Sir, can you explain monoclonal antibodies?
Of course. These are lab-made antibodies targeting specific molecules like rituximab, which is anti-CD20 in lymphoma, or infliximab, which is anti-TNF-alpha in rheumatoid arthritis. Let's recap. First, antibodies are Y-shaped proteins with variable and constant regions. There are 5 classes: IgG, IgM, IgA, IgE, and IgD, each with unique functions. They neutralize, opsonize, and activate complement. Clinically, they are vital in diagnosis, therapy, and vaccines. Any final questions?
Sir, how do B cells decide which antibody to produce?
Smiling. That's a topic for our next lecture. B-cell activation and somatic hypermutation. For now, review today's notes. See you next time. Smiling. That's a topic for our next lecture. B-cell activation and somatic hypermutation. For now, review today's notes. See you next time.